Maryke Appel from Kapa Biosystems, who will be giving a talk on the evolution of library prep, selecting for both quality and speed.
Thanks, George, for the introduction, and to the organizers for the opportunity to speak here today about the evolution of NGS library construction. Our activities in this field have primarily been focused on two goals. First, the performance improvements needed to expand the pool of samples, low input and challenging samples that can be successfully processed in standard sample preparation pipelines. And then secondly, workflow improvements needed to support higher throughput and faster turnaround times. Our efforts over the past 4 years have resulted in intangible improvements to Illumina sequencing pipelines. This is an example from TGen in Arizona, where the introduction of Kapa's first improved library preparation kit reduced the input requirement for the exome workflow from 3 micrograms to 200 nanograms. And then with the introduction of our streamlined hyperprep kit, that input requirement was reduced another 4-fold while cutting the library preparation time in half. And so in the next few minutes, I will explain how a better conceptual understanding of library construction and a systematic approach have allowed us to achieve these improvements. And then I'll also introduce our latest and most exciting library preparation workflow, which we call HyperPlus. So HyperPlus combines a robust and low bias enzymatic fragmentation reagent with the Kapa hyperprep chemistry in a single tube fragmentation library construction workflow that really represents the ultimate combination between performance and speed. And so where did all of this get started? So the first step in our conceptual evolution of library preparation was really to answer the right question. People who make libraries every day are pretty much focused on input and output, and very rightly so. And so for them, the primary question is, can I make a library with the amount of the type of material available to me? Can I make a library that's appropriate for the sequencing application? What we came to understand is that it's not about whether or not you can make a library, but how do you know that you're making the best possible library from the available material? In other words, the library that's most diverse and results in the highest and most uniform sequence quality or coverage. And so to answer this question and really effectively drive product development, a good proxy for library quality is needed. And we know that from our work with library amplification that yields of final library aren't good predictors for sequence quality, because for a variety of reasons that I can't explore right now, there's a disconnect between the input into library construction and final library yields. And so if you dissect the sample preparation process into its functional units, it's clear that the core library construction process, so the process in which adapters are added to randomly fragmented DNA, that's really the process that determines the number of unique molecules that's available for sequencing, while library amplification only really generates duplicates. And so to really evaluate the intrinsic efficiency of a library construction workflow, it's not the final yields of library, but really that intermediate step or post-ligation library yields that becomes a good indicator of what's really happening. This, of course, assumes that library amplification is well optimized and performed with a low bias enzyme. And because absolute yields of post-ligation libraries aren't very useful to compare workflows across sample types and inputs, we defined this term conversion rate, which is essentially just the percentage of input DNA converted to a data-ligated library. And using this sort of normalized metric, we were able to benchmark performance, and that really kind of guided our efforts towards making both performance and workflow improvements over the last few years. When we first started focusing on library construction, conversion rates were actually alarmingly low. And so this example, which gives the average conversion rate for libraries prepared from 1 microgram inputs of bacterial DNA for whole genome sequencing, was fairly representative of what could be achieved with sort of the state-of-the-art chemistry at the time. With our first DNA library preparation kit, in which we pretty much made a few changes to reagent formulation and we added 1 extra cleanup to a workflow, we achieved more than 10% conversion rate for the same sort of input and sample types. And so when we start plotting the evolution of library construction on a chart of some sorts, expressed as the maximum conversion rate for 1 microgram input of good quality DNA, it sort of starts looking like this. So we're starting with a baseline less than 1% material converted into usable library, to over 10-fold improvement just by making some reagent changes and changing the workflow a little bit. And although this was a significant gain, we knew that this process was still very, very inefficient. And one of the biggest problems with the conventional workflows at the time was just the huge loss of DNA as a result of the physical transfer between tubes in which end repair, A-tailing, and ligation were taking place. And so we addressed this by incorporating the so-called with-bead strategy that was published by Fisher et al. From the Broad and Foundation Medicine early in 2011. This strategy for the first time allowed the core library construction process to be completed in a single tube. Not only did this make the process a lot more easy to automate, which is really important to support the higher throughputs that we see in NGS, but it also really improved the efficiency of library construction, now allowing successful library construction from low input and challenging samples such as FFPE. While we implemented the strategy, we also looked at other key library construction parameters such as adapter concentration, cleanup, size selection, and amplification parameters. And the result of this comprehensive effort was another 4- to 5-fold improvement in library construction efficiency with a so-called Kapa with bead or HTP library preparation kit. And this tracked down for the first time to about 100 nanogram inputs. And so the workflow and performance improvements embodied in the Kapa with bead protocol became enabling for clinical sequencing pipelines designed to process low input and challenging samples such as circulating tumor DNA and FFPE samples, as exemplified in these publications from Stanford and the Broad, published during the course of 2013. Although we had achieved a lot, we also knew that there was a lot of room for improvement. And so in terms of workflow, we knew that we needed to start streamlining the workflow to support higher throughputs, as others had already started doing at the time. And then in terms of performance, a collaborative study that we presented at this meeting last year on FFP library construction showed conversion rate to be the bottleneck in FFP library construction. Because conversion rates are so much lower for FFPE than high-quality samples, you have to amplify a lot more, and that leads to the high duplication rates that are typical for FFPE. As we started streamlining the protocol, we soon realized that we were pretty much faced with this dilemma of essentially divergent evolution. So as we just took the cleanup steps out of the with bead protocol, it put us back a whole generation. And so from 40% conversion rates, we went down to less than 10% on 100 nanograms of DNA. And so the challenge really was how to achieve cumulative improvements. How do you streamline the protocol but retain the gains in performance that we've achieved with the with bead protocol? And so we started tinkering with the chemistry, not me, the good guys that's in the audience here, through formulation after formulation and several prototypes, until we eventually arrived at a chemistry that gave us equal or better performance than our with bead protocol and better than any other streamlined kit available at the time. And so the result of this effort was the Kapa HyperPrep kit that we first introduced at this meeting last year, and it was officially launched in April. And so this kit combines end repair and A-tailing in a single reaction. It eliminates cleanup steps and thereby cuts library construction essentially by half. And so you're able to make libraries from mechanically sheared DNA in 2 to 3 hours, depending on whether you're doing amplification or not. What was even more significant is that hands-on time or handling time was reduced by 2/3, and so it made the protocol even easier to automate, but just the simplicity of the protocol increased consistency and reproducibility in manual settings as well. And so a combination of this really simplified protocol and the novel chemistry led to even better library construction efficiency. So these are some samples from our 2014 FFPE study, where with the with bead protocol and high-quality control DNA, conversion rates were in that range, expected range of 25 to 40%, whereas conversion rates for FFPE samples were in the range of 5 to 10% and quality dependent. And with the HyperPrep chemistry, conversion rates became comparable to what we achieved with good quality DNA processed in the same workflow. This obviously has huge implications for lower, less amplification, lower duplication rates, better coverage, the sensitivity of clinical assays. But it also started sort of lowering the bar, so lowering the requirements in terms of input and quality for samples that could be successfully processed in standard pipelines and with predictable success rates. And so on our evolutionary chart, pretty much looks like this now. With the HyperPrep kit, we could get up to 60% of input DNA converted to adapter ligated library down to about 100 nanograms, but also quite significant in the lower input range, so in the 1 to 10 nanogram range, where ligation-based library preparation is definitely challenged. we were achieving conversion rates up to 10%, which was significant. And so at this juncture in the evolution of library prep, what remained for us to do is pretty much best summarized by a quote from a French aristocrat and aviator who said, "Perfection is not achieved when there's nothing more to add, but when there's nothing left to take away." And so having addressed most of the pain points in the Illumina sample preparation pipeline, a robust and accurate library quantification, sample quantification and QC, and library amplification, which we didn't get to speak about today, now the core library construction process, really the only process that was left to tackle is fragmentation. And so in line with what we've been doing for the last few years, we realized a few things. That whatever fragmentation solution we came up had to be enzymatic to support high workflow, high throughput. Like the Kapa hyperprep kit, it had to be versatile, so be able to support library construction from different sample types, different inputs, and for different sequencing applications. And it had to build on the performance gains that we had achieved to date. In other words, support really high library construction efficiency and not introduce any sources of bias. And so we're really excited to announce that such an enzymatic fragmentation reagent is now available. This is just some bioanalyzer profiles for DNA, so 100 nanogram inputs of different types of DNA, from complex human genomic DNA to PCR products, sheared for different amounts of time with our enzymatic fragmentation reagent. And you can see the profiles are very phenotypically very similar to Covara shears, and that the mode or average fragment size is very consistent, irrespective of the length or the complexity of the input DNA. And so with this reagent, you can now shear anything from a nanogram to a microgram of DNA of any kind of type in any fragment sort of range that supports Illumina library construction, and hopefully even beyond that. What's not really that exciting is, the fact that we built a really useful enzymatic fragmentation reagent isn't really the most exciting part of it. The most exciting part of it is that we've been able to fully integrate it into the hyperprep chemistry. And so now you can start with your DNA in a tube and do fragmentation all the way to the post-ligation cleanup in a single tube in pretty much the same time in which you did the library construction process in the past. So anywhere between an hour and a half and 3 hours, depending on what your mode fragment size is and whether or not you want to do amplification. And this workflow retains all the flexibility of the hyperprep workflow in terms of input, sample type, sequencing applications, adapter design, barcoding strategy, whether or not you want to do amplification and whether or not you want to do size selection. And a combination of eliminating even more of the sample losses associated with the physical transfer from mechanical shear DNA into library construction, and also a synergistic effect between the enzymatic fragmentation chemistry and the hyperprep chemistry has led to exceptionally high and surprisingly high library construction efficiencies. And so now from 100 nanograms upward, we approach library construction efficiencies of almost 100%. And so on our evolutionary chart, we've literally now kind of achieved the holy grail of library construction, where you can start with a microgram of DNA and end with a microgram, almost a microgram of DNA without doing any library amplification. And this obviously opens huge doors for PCR-free whole genome sequencing, PCR-free targeted sequencing, and other emerging applications where really high library construction yields are necessary. One really good example of that is so-called oligospecific sequencing that was developed in Hanley G's lab at Stanford. Eric Hopmans has a poster on that showing that with this amplification-free technique, you can achieve really, really high coverage depths and start detecting mutations present at frequencies less than 1%. Again, it's not just at the high input range of the spectrum that we've achieved huge advances, but also on the low input range. And so from a nanogram of DNA, we can now get almost 20% of that into usable library. And it just means that there's a huge pool of samples that previously or with other chemistries will just fail library construction entirely, can now be processed, and also in a way that doesn't require so much amplification that coverage ends up being compromised. And thanks to our early access collaborators for all their hard work and testing, we have a wealth of data available that essentially shows two things. First of all, unanimously higher library construction yield. So this is just one example, and we have a ton of these on a poster in our suite for both high quality and FFPE DNA and different types of samples. So unanimously, everybody sees higher library construction yields when you use the same hyperprep chemistry, so basically our best library preparation chemistry to date, with enzymatically fragmented DNA versus Covara shear DNA. Because you have higher yields, you have higher library diversity, means you can do less amplification, leads to lower duplication rates and increases in coverage, which I'm not showing here. The second thing that everybody showed is that this doesn't seem to come at any cost. So there's no trick here. There's no cost that we're paying. So if you look at other sequencing metrics like genotype concordance or specificity of SNP classification, we see no difference between basically the molecular nature of libraries made from enzymatically fragmented DNA or libraries made from Covara shear DNA. And so in the last few minutes I have available, I'm going to address the one big question that everybody's skeptical about, which is obviously fragmentation bias. So as I was telling people about this over drinks the other night, they're like, yeah, we've heard this before, we've tried this before. And so obviously, I mean, even we were skeptical about it, that it was possible to overcome that fundamental issue. And so start site bias is one way to look at fragmentation bias. And so this is data from exome capture experiment, and we've done the same for microbial whole genome sequencing. If you look at sort of the nucleotide distribution or content around the start of the alignment, the read alignment, you can see that there's a little bit more uneven distribution for enzymatically shear DNA than Covara shear DNA, but it looks a hell of a lot better than libraries made with tagmentation-based technology. And the great news here is that this little bit of start site bias doesn't seem to affect overall sequence quality. So these are the coverage uniformity plots from the very same experiment, so human whole exome capture experiment. And so for this type of plot, what you would like to see is a very narrow distribution at the targeted coverage depth, the desired coverage depth. And two things are very obvious here. So here the coverage profiles for libraries again make from enzymatically fragmented versus Covara shear DNA with the hyperprep chemistry, and there's no significant difference between the two, and a fairly tight distribution around 20x. If you compare this with DNA made with tagmentation technology, you see two things. So you see these coverage hotspots where you're basically just wasting sequencing reads because all the easy kind of bits are just covered at much, much higher depth than you need. And then you see these sort of areas where there's very low and lumpy coverage. And the result of this is a lower mean coverage. And so you have to do a hell of a lot more sequencing to raise that mean coverage to the levels that we can achieve with ligation-based chemistry. And even if you did all that much sequencing, you'll never change the shape of that curve because of the intrinsic biases associated with the fragmentation technology and also the fact that amplification is mandatory in these workflows. And so I'll finish with one final data set. This is for libraries made from 1 nanogram of bacterial DNA for whole genome sequencing. In this case, we again used our gold standard, Covara shear DNA with the hyperprep chemistry, or the hyperplus, which is the hyperprep with enzymatic fragmentation, tagmentation. And then we included another competitor, so this is another enzymatic fragmentation reagent plus a streamlined library preparation kits. And so, with the exception of the Covara sheared workflow, all three of these are similar in terms of speed and convenience and hands-on time, but we see huge differences in sequence quality in the end. So in these type of GC bias plots, obviously, you'd like to see a line essentially at a normalized coverage of 1, a horizontal line, because you'd like the coverage for each GC bin to be similar to the coverage for the mean. And so what we can see again, whether you shear enzymatically or with Covaras, there's no huge difference in the data, and that kind of line approaches normalized coverage of 1, especially kind of in the more difficult areas, where with the competitor chemistries, there's huge underrepresentation of the difficult areas, and so requiring a lot more sequencing to get them to the same coverage uniformity. You see the sort of same pattern when you look at an organism with a GC-rich genome. So here the mean is around 70, it's just about 70, and you can again see, so here's enzymatically fragmented DNA in Covaras. It's a little tougher, it's not perfect, but approaching a lot more sort of uniform coverage than some of the competitor chemistries. And it's best exemplified with the HD-rich, so this is Clostridium that has 30% genomic GC content. We have a virtual perfect distribution with both enzymatically fragmented Covara shear DNA, and we see these huge areas of overrepresentation of easy stuff and underrepresentation of difficult sequence, resulting in, again, of having to do a lot more sequencing to get to the same coverage uniformity. And so to conclude, through essentially a really systematic approach, we've been able to achieve cumulative improvements to both performance and workflow for Illumina library preparation. It's not only helped us to support higher throughputs and making things easier and faster, but really expanding that pool of samples that can be processed successfully and with predictable success in standard preparation pipelines. So without having to bin samples or separate them or pre-treatment, just kind of pushing them all through the same system. We now are applying some of these principles to RNA sequencing as well. Unfortunately, I didn't have the time to talk about it, but we've got all the scientists in the suite, and they'd love to talk to you about some of the things that we have in the pipeline there. And then to summarize, HyperPlus workflow combines low-biased, robust enzymatic fragmentation with really, really high library construction efficiency and our high-efficiency, low-bias amplification enzyme to really create an industry-leading solution that has the speed and convenience of fragmentation-based library prep, but with significant improvements in terms of sequence quality, robustness, flexibility, and ultimately sequencing cost. And so I'd finally like to acknowledge all the guys that build these wonderful products, everybody that kind of helps to support them and bring them out to you and have in some way or another contributed to this presentation. Our commercial collaborators who's helped us over the years automate all these wonderful kits and help us integrate them into target capture solutions. But really the people who we want to thank are you guys, our customers, collaborators. Without your inputs and suggestions and feedback and all your test results, none of this would have been possible. And so we really have been standing on the shoulders of giants here. And we really want to thank you, and we want to dedicate this really, really exciting new chapter in the evolution of library preparation to you guys. Thank you very much.